Oh, so I'll start. Hello, everybody. It's nice to speak with you today. The title of this talk is Pathway-Based Approaches for Disease Identification, Novel Approaches and Pitfalls. And I would like to recognize some of my co-contributors who include Ivan Gorloff here at Dartmouth, Heike Bickerböhler in Germany, Mamia Xiong at UT Health Science Center in Houston, and Siddhartha Kar at Cambridge, and Saskia Freytag also in Germany at Göttingen. Before we get started, I thought I would just mention as an example an application of a pathway-based approach, which was recently published, and I'm going to push this link out to you, which is a Nature paper. It's discussing a sort of integration of multiple studies, genome-wide association studies, that identified novel variants and then use those variants to explore pathways that influence susceptibility to disease, and subsequently identified compounds that have worked for treating rheumatoid arthritis, as well as suggesting new compounds that may be helpful. And so the idea of pathway-based approaches is that when we perform studies such as genome-wide association analyses, we identify many variants that are relevant to disease causation, but the integration of these findings may be complicated, and additionally, many variants that we identify may not reach the stringent levels of significance that we require, and the pathway-based approaches may provide more information about the variants that influence disease susceptibility, while also integrating the information into more biologically meaningful results. So going back to the presentation, I'll move on to the next slide. So in order to move back, since I pushed this out to you, you'll have to click on the tab at the top that shows the presentation. So I've developed this presentation as a set of questions. The first question is, what is a gene pathway? And one definition would be that it's a set of genes that jointly coordinate a cellular process. One example would be the cytokinesis pathway or tricarboxylic cycle. These kinds of pathways are cataloged in several different resources. One of them is the Gene Ontology project, and that project organizes genes into certain attributes such as the cellular component, molecular function, or biological processes that the genes influence. There's a software program called Amigo that's helpful for people that would like to retrieve data. And this software draws in data from multiple sources, such as UniProt, which provides information about interactions among genes at the protein level. When you do queries from Gene Ontology, you can control what information you will be retrieving. And this slide is showing you the different aspects of the retrieval that you can control. So that's the nice thing about the Genome Ontology environment. Additional resources for identifying pathways that are commonly used includes BioCarta, which has user-supplied interactions mostly derived from proteomic studies. There's the KEG, Kyoto Encyclopedia of Genes and Genomes, which is manually curated from the literature and describes gene-gene interactions. So this is very popular because it's been carefully evaluated for consistency and also provides nice graphical interfaces. There's a couple of available software programs. Ingenuity is available and provides pathway information. It's maintained on a central web server, so it's updated regularly by their company. Pathway Studio also does something similar, but it distributes the pathway out to the users, and so it can be more rapid computationally for people that are doing a lot of work sort of online. So here's a typical example of a pathway diagram from the Kyoto resource, and I'm just showing this as an example. You have interactions among multiple genes, and then you also have, in sort of circles here, additional places where additional pathways interact with this particular one. So pathways are not independent. They form a complex network among pathways, and so that's a little bit of a complexity in the analysis that we perform. An additional kind of approach is text mining. So this is not really a pathway-based approach, but it's a way of identifying relationships among genes. One example is called GRAIL, and it uses text mining of published papers to identify relationships among genes that have been presented in the literature. So the advantage of this is that it's quite automatic and updates very quickly, and it does provide new insights. The disadvantage is that just because a couple of genes are mentioned in a paper does not necessarily mean that they have meaningful interactions at the protein level. So it's sort of a useful resource, but more limited than the other pathway-based approaches I described previously. Now the question is, what is a gene? So we expect to include components of a gene that code for the protein. Most genes have non-translated components that affect the tissue-specific expression, and so they are relevant to how the gene influences a disease development. Many genes have multiple transcripts, and some genes, like the UGT family, include many members that have shared exons. So sometimes the definition of what a gene is a bit complicated, and that becomes relevant, especially in studies from genome-wide association analysis, because we typically define a region as being the gene, but in some cases the region can be multiple different genes, and so there's a lack of uniqueness in some cases. And many genomic regions include RNA products, and so our definitions are getting broader as we move forward in time. Standard annotation approaches like Gene Ontology may not well capture information about some of these non-coding sequences yet, and many genes are not yet mapped to pathways, so that's a limitation of a pathway-based approach. And how a pathway approach has been used, I discussed that briefly in the beginning. These arise from gene expression paradigms, usually in the past, for which gene expression arrays provide information about RNA expression levels. RNA expression panels typically have about 20,000 transcripts, and the pathway-based approaches were most popular, or are most popular, for Gene Set Enrichment, which evaluates departure of p-values from an expected uniform distribution. The GCTA approach for expression analysis uses an enrichment score in which let's say you're studying N genes in an expression study. You rank order those according to expression, and you would form a set that are in a pathway, and then you identify the correlations of the expression patterns with the phenotype of interest. So that's the RJs listed here. So, for example, that might be correlation between expression and case control status. And then one evaluates the fraction of genes in the pathway weighted by their correlation against the fraction of genes not in the pathway, and form a statistic and elements of this statistic. So S is the members of the pathway, C is the phenotype, and P is a kind of a weighting criterion. And when P equals 0, this is a Kolmogorov-Smirnov test, and when P is 1, then it's weighting by the correlations normalized by the sum of all correlations in genes. So anyway, this provides a way of prioritizing how strong the evidence is for the pathway being involved versus against evidence for that. And so in the initial paper, there's a few examples. The left panel, S1 here, was looking at expression comparing males and females, and those were X-linked genes. So you expect that there would be upweighting for more genes highly expressed related to male-female differences. So that occurred in S1. S2 is sort of a null, and S3 shows sort of complicated upweighting of a pathway. So that's sort of the example from expression arrays, and now we have the problem of how do SNPs relate to genes. So the usual approach that we typically apply sets a window around the coding region of genes, for example, a 50 kilobase window. And then multiple SNPs occur within a gene, and we usually use single nucleotide polymorphisms for this analysis. One of the complexities here is that large genes typically include more SNPs, and SNPs that affect gene expression may reside either inside or outside the coding region. We hope that the 50 kilobase window that we typically apply includes enough of those SNPs, but in some cases that may be problematic. An example is CMYC. And closest SNPs to a gene might affect some other nearby genes. So that, again, that's another limitation of this kind of approach. As we go forward in time, we may get better at identifying which SNPs are influencing which genes, and so we're doing sort of as well as we can by this window approach, but it's a limitation at the moment. So how do we use SNPs to represent pathways? So in the Gene Set Enrichment approach, the way this has been applied is to use the most significant SNP in a gene as being the one that represents that gene. Again, larger genes have more SNPs, and so the p-values relate to the number of SNPs that were studied. And the immediate concern is this might lead to more positive findings for larger genes. One way to manage this is by a permutation procedure. You could also use the sum of p-values across SNPs, and that will not cause a problem of inflation with larger genes, but it might lose some power if multiple SNPs are unrelated to the disease risk that's studied. And you would need to allow for some kind of correlation among the SNPs. So you could also try doing empirical evaluation. There's a nice software called SCAN, which helps you to pull up expression levels of SNPs related to genes of interest, and so I presented that here for you to look at. So here's the Gene Set Enrichment for GWAS. There's a seminal paper by Wang et al. They actually applied this approach to the Parkinson's disease example, and they used the GCTA approach I described earlier and applied the most significant SNP in a gene. This method worked quite well in this example. And this is sort of another application to macular degeneration, again, showing that it worked fairly well in this example. Another kind of approach that you might take, because they're could be many different genes involved in or suggested as playing a role. You could apply penalization approaches to limit the number of genes that you consider. So there's this nice paper by Chen et al. As examples, we've also applied genome-wide gene and pathway approaches. So my colleagues and I developed a method, another set of methods that manage the correlations among SNPs. Rather than picking the most significant SNP, we used a combination of all of them. And the nice thing about these studies is they took advantage of multiple data sets in rheumatoid arthritis. So one could build a model in one data set and validate it in the next. There's also a nice paper referred in the last slide by Carr et al. To primary biliary cirrhosis. In this application, there were two different tests that were developed. One was a linear combination test, in which the p-values at each position were summed together and adjusted for the correlation among the SNPs. So that removes any concern about length bias of the SNP or the pathway influencing the number of significant results that one obtains. An alternative approach is the quadratic test, in which the p-values are used directly and adjusted for the correlation among the SNPs. And when these methods were applied, in the linear combination test, we obtained a large number of interesting SNPs. Many of these were in the HLA region, so of course these are likely to be correct. And PTPN22, which we know is a gene related to rheumatoid arthritis, also shows up in the top set. When the quadratic test was used, a large number of pathways were identified, and these make some sense. Several new pathways were identified by analyzing these data. And if you compare the pathways between the two different data sets, the Wellcome Trust and the North American Rheumatoid Arthritis Consortium that rose to the top, same pathways were identified. Individual genes that are included in the pathway were not necessarily the same. So in this example, the red marks are the significant genes in the pathway, and on this slide we show the results for the Wellcome Trust finding, and the next slide we show for NARAC. So there were different members, but the entire pathway shows up, and so that's kind of the value added of the pathway-based approaches, that you don't necessarily have enough information to pick up all of the gene signals in a pathway, but by coordinately analyzing all those genes, you can still pick up the pathway as playing a relevant role for disease causation. So I separately applied these methods in a genome-wide association study of melanoma that I conducted with my collaborators at MD Anderson and additional collaborators in the UK and Australia. And so this is the genome-wide association study results showing all the different significant findings across the genome. So we took those results and applied the LCT and the quadratic test that I described in a couple of slides earlier. And the significant pathway, many significant pathways were identified by the quadratic test, which I thought was very exciting, but on further evaluation of these pathways, noticed that some very lengthy pathways were rising to the top. And although the neuroreactive ligand receptor interaction pathway is kind of interesting, it's not something that I necessarily would have thought of as my top hit. And because I was a little anxious about that, we decided to permute the data. So we took the the cases and controls in the original data set and permuted those, swapping the labels. And when we did that, we found, unfortunately, a very strong correlation between the size of the number of genes in a pathway and the significance, and even the number of SNPs in a gene and the significance. This is something that you really don't want to occur in a pathway-based approach, because it raises a concern that you're finding false positive findings among the true positives. And we didn't see that in the LCT application. The original paper didn't show any bias, and so I think the particular issue here may have to do with the R code that we're using for this application. In general, I think I'm just trying to point this question out as something worth evaluating. If you have a length or a pathway bias in your results, it's worth taking a second look. So this just shows you sort of the correlation. So in the LCT approach, there was no correlation. In the QT approach, in this application, there was a correlation. Sort of moving forward in time, we've been working with our collaborators in Germany. They've been driving the development of logistic kernel machine test approaches. So these approaches first adjust for covariates. So that's what these regression coefficients, beta 1 to beta m, are doing. And then there's a linear kernel, so that's this H component, which seeks to identify the set of variants that optimally separate cases and controls. And then so this is a semi-parametric approach to identifying genes, and then SNPs within genes, and then genes within pathways that relate to disease causation. And to perform the test, one forms a quadratic form, which turns out to be a sort of complicated mixture of chi-square variables. Fortunately, the literature on pathway-based, kernel-based methods is well developed, and methods for identifying the correct distribution of chi-squares are well established. So this method could be applied. And so the things that we need in a kernel-based test or any pathway-based test with SNPs is that there's size invariance, and we correct for the correlation structures among the SNPs, and finally that we incorporate the pathway structure. So to do this, Saskia Freytag and Heike Wickeball developed size invariant multiplicative Euclidean distance kernels, and the kernel includes this Euclidean distance measure. It corrects for the number of genes in a pathway. It groups the SNPs with regard to their gene membership, and then it corrects for the number of effective SNPs in the gene by identifying the number of effective tests that were conducted. And then one needs to construct a network-based kernel. So first that's done by mapping SNPs to genes, and then one also maps the genes to the pathways and adjusts for the correlations among the genes within the pathway. And so in this application, a similar analysis was conducted of the North American Rheumatoid Arthritis Consortium data, and 3 different methods were applied: a linear kernel machine test with a linear kernel alone, one with a network-based kernel, and then one using Gene Set Enrichment. And we felt that the network-based kernel provided the most succinct sort of characterization of the pathways that were identified. And in this figure, I'm giving you the two references for this method. And some of the top pathways from analysis include type 1 diabetes, systemic lupus, rheumatoid arthritis, which we expect, viral myocarditis, and the phagosome, which is probably a new network for rheumatoid arthritis. So there were some novel findings from this analysis. Future directions would evaluate evaluating the behavior more completely, comparing this linear kernel machine test to some other methods, integrating gene and pathway-based approaches from SNP GWAS studies with, let's say, the Cancer Genome Atlas. So one of the sort of challenges that we have in the cancer world is that we have some very large and excellent studies being conducted to identify genetic susceptibility for cancer, and then we have a whole separate set of analyses being conducted to explore the changes that occur in the somatic tissues, and these two sort of approaches aren't very well integrated. So if we could further integrate those, that would be a value added. And then further exploration of application of methods using some new approaches. There's a GCTA approach which can be used for genome-wide association studies to first identify the most significant SNPs in a region, then remove any additional SNPs that are being identified because they're in this equilibrium with the most significant SNP. So condition on the most significant SNP, then identify all the additional SNPs that remain after that conditioning. That will result in a more parsimonious view of those SNPs that are relevant for the relationship of genes to disease causation, and trim the number of genes that are being nominated for inclusion in the pathway analysis. And we've done some work on this kind of approach, finding that it does certainly lead to a much reduced number of markers and genes to consider in the pathway studies. So I think I will stop at that point. I think I'm sort of a little in good shape here for time, and I will ask questions. I'm not sure I see any, but let me click on my buttons. So I don't have any questions right now. If there are any, I will answer them, but otherwise I will just be finished. I'll wait a second or two. I want to thank my collaborators for their input, and this is a very interesting area that's continuing to develop. There are massive amounts of genome-wide association studies that have been very informative, and being able to translate these into clinically meaningful applications is sort of our goal. I would like to point out that I saw a very interesting, let's see, let me just say, I saw a very interesting recent result that a novel therapy for alopecia areata was developed from observations that the JAK-STAT pathways involved in alopecia areata. So that led to application of drugs that target that pathway, and there do seem to be novel approaches that are being for clinical use from genome-wide studies. So I see a note that says only 3 last slide was in the content. Will you change that? Yeah, I will change that. I'm not sure I understand the question. So I guess that's all that. I guess that's the end of my presentation.